Our last lecture is going to be taking the idea of getting from a gene that you find in DNA and getting that to a protein. By now you guys know that DNA is what makes us what we are. We are programmed, our characteristics, our personalities come from our DNA, so they lead to the specific traits about us. Now, what gives us those specific traits are actually proteins. So the idea is to figure out, well, how do we get from DNA, which is the recipe for what we are, to building the proteins, which is what we actually are. So proteins are those links between the genotype, which is your genetic makeup, and the phenotype, which is your physical expression. Now, you're going to learn the two processes that we have to go to get from DNA to the proteins at the end of the day. So you have the first one called transcription, and this is going to be going from DNA to an intermediate called RNA. And then there's translation, which is taking that RNA and then converting that into the idea of a protein. So we're going to do an overview of these two processes, and then we're going to do them in detail. So in our DNA, we have those genes, which are short sections of the DNA that code for a specific trait, and those specific traits are dictated by the proteins we build. However, the DNA does not directly go to building those proteins. DNA contains the information, and we're going to basically build that into a copy of another type of nucleic acid, because remember, DNA is deoxyribonucleic acid. We're going to build that into a second type called RNA, which is ribonucleic acid, and then that will direct the synthesis of the protein. So RNA, ribonucleic acid, you have to know the differences between DNA and RNA, and I'll give you three major ones. First, different sugar. If you guys remember, this is something you learned before. In DNA, you have a sugar called deoxyribose. I should finish saying that's a sugar. And then over here in RNA, we have ribose. So we have two different sugars. DNA has deoxyribose and RNA has ribose. So that's one difference. Another difference is the overall shape. So shape is a difference. If you remember DNA, it's a double helix. When you look at RNA, it's a single-stranded molecule. So DNA is a double helix, which means you have two sides basically helically bound like that. And then RNA is just a single-stranded molecule. So that's the second difference. Different sugar, different shape. The third difference is there's actually a different nucleotide. So remember, DNA and RNA are nucleic acids, and nucleic acids are built out of nucleotides. So in DNA, you have adenine, thymine, guanine, and cytosine. In RNA, there is adenine, there is guanine, there is cytosine, but there is no thymine. There is a different one instead called uracil. So U and T are different. So U in RNA for uracil, T in DNA for thymine. So thymine in DNA, uracil in RNA. So different sugar, different nucleotide, different structure. The first process we're going to talk about is transcription. And once again, right now is going to be in general. So in general, you have to know transcription is taking a DNA molecule and converting that into an RNA molecule. Specifically, the type of RNA we're going to build is called mRNA, and that stands for messenger RNA. Because if you really think about what's happening here, we have DNA. DNA is in the nucleus of the cell, and in that nucleus of the cell, we're keeping DNA there to protect it. So we don't want DNA to leave the cell's nucleus. We're going to send a message out, and that's called mRNA. mRNA is what carries the information to tell the ribosome how to build the proteins. Because remember, at the end of the day, we're building proteins, and if you're building proteins, that happens at an organelle called ribosomes, and you guys learned that back in the middle of the semester. So there's a few general things to know here. First of all, you have transcription, which is converting DNA to RNA. Specifically, you could say mRNA, and this occurs in the nucleus. Now, how would you already know it occurs in the nucleus? Because I just told you DNA is in the nucleus and it's not coming out. So if you're going to build mRNA from DNA, it has to occur in the nucleus. So DNA to mRNA is occurring in the nucleus. And that mRNA is the message going from DNA to the ribosome to dictate how to build the protein. Now, in general, just so you guys know why it's called transcription, the idea of transcription is basically what you do when you take notes. You write something down. So you're just rewriting what I'm telling you or rewriting what I put on the screen. Now, when we say DNA, and I say what type of molecule is it, you should know it's a nucleic acid. When we look at RNA, what type of molecule is it? It's a nucleic acid. Well, aren't they both nucleic acids? Isn't that the same language? If you're just writing it down, hence the term message, you've transcribed it. That's why it's called transcription. It's basically just rewriting the message. The second process is called translation. And translation is when you go from an mRNA, so remember that's an mRNA that we're working with, to the actual protein. So this is going to occur at the ribosome because if you're building proteins, that occurs at the ribosome. We're going to take the mRNA, it's now going to leave the nucleus, it's going to go out to the ribosome and then dictate how to build that protein. And of course, proteins are built out of amino acids. I taught you at the beginning of the semester what the building blocks of molecules are. So RNA is a nucleic acid built out of nucleotides. Now we're building proteins made out of amino acids. Now think about that for a second. Why do they call this translation? We go from RNA, and RNA is a nucleic acid, and that's built out of nucleotides. We're going to a protein, and they're built out of amino acids. Well, think about that term translation. When you translate something, don't you change the language, going from German to French or English to Spanish? Well, if you go from a nucleic acid to an amino acid, then you're changing the language. That's why it's called translation. So transcription, DNA to RNA, that occurs in the nucleus. Translation, RNA to a protein, that occurs at the ribosomes. So I mentioned earlier that DNA is not what directly dictates how to build that protein. DNA has the instructions, but it doesn't go out and do it. Why do we use that RNA as the intermediate step? Wouldn't it be easier just to send DNA directly out? Well, it might be more straightforward, but the problem is it's too risky. If DNA leaves that nucleus and goes out into the cytoplasm of the cell, there's a high probability that it'll become damaged. There's a lot of chemical activity going on in the cytosol, or the cytoplasm, so we're trying to keep the DNA safe. That nucleus, where you have DNA, is like a safe. You're locking it away in there so everything's okay. Now the other thing is, if you send that DNA molecule out, you can only read it one at a time. But if we make mRNA copies, we can make endless copies, which means we could be building all these proteins all together at one time. It's like your note packet. If I come in with one note packet and I said, this is the note packet, everybody has to share it, and you guys had to take turns passing it around, studying from it, that just wouldn't make sense. But if I take what I wrote and make copies and give you each one, you can all use it at the same time. So it's much more efficient that way, and you're protecting the DNA. That's why we're using RNA as that intermediate step. So we have our nucleus inside of the cell, and that's where our DNA is, that double helix. And what we're going to do is we're going to take DNA and we're going to convert it into an mRNA. That's the process of transcription, and that occurs in the nucleus. mRNA leaves, it goes to find the ribosome, and we're going to build that into a protein. So remember, polypeptide is another name for a protein. So when you go from mRNA to a protein, that's called translation. So transcription, DNA to mRNA, translation, mRNA to a protein. Translations out in the cytosol, specifically at a ribosome, and transcription is in that nucleus. In the process of getting from DNA to a protein, we have to figure out how that language is understood. You guys know that there's 4 different nucleotides. However, when you look at proteins, there's 20 different amino acids that are needed to build all the known proteins. So the problem is, how do we get from 4 to code for 20 different amino acids? Well, let's look at the idea here. If one nucleotide coded for each amino acid, so let's say you took a thymine and that coded for a phenylalanine, and an adenine coded for a proline, and a glycine was coded by cytosine. Well, at the end of the day, you would only get 4 amino acids. That's nowhere close to the 20. But what about if we read 2 nucleotides at a time, like TNA or G and C or AG or CG? Well, if we read 4 at a time, we have 4 bases, 2 together, that's 4 raised to the second power. That'd give you 16 different combinations for the amino acids. The problem is that's still not 20. So reading a nucleotide 1 at a time doesn't work. Reading nucleotides 2 at a time doesn't work. What about if we read it as a triplet, 3 at a time? Like we could read A, C, A, or T, C, T, G, C, or C, C, G, however you want to combine it. So there's 4 bases. If we're reading them 3 at a time, 4 raised to the third power gives us 64 different combinations, and that's more than enough for the 20 amino acids. And this is actually how the information flows. When you read DNA, you read it 3 at a time. That's called the triplet. When you read the RNA, you still do the same thing. Read 3 at a time. That's called a codon. So triplet is a term that you use with DNA. When you hear the term codon, that is a group of 3, and that you use with RNA. So triplet in DNA, codon in RNA. So you follow this group of 3, and that's how the information flows. When we look at the idea of transcription, you guys realize you're going to go from DNA to RNA. The problem is DNA is a double helix. It's a double-stranded molecule. RNA is a single-stranded molecule. So we only need to read one side of that DNA. The side you read is called the template strand. Now, you have to know what the template strand is. Simply put, it's the DNA strand that you're copying. However, you don't have to copy both sides, and you're not going to have to identify which side's which. It's kind of written in the code itself. But the template strand is the side of the DNA that you read from. So the mRNA nucleotides are going to be matched up one by one with the DNA nucleotides. And once again, you're going to use that base pair rule. Remember how in DNA, an A goes with a T, and a G and a C go together? We're going to have a very similar thing in mRNA. If you have an A, you're going to go with a U. There's a difference. If you have a G, you're going to go with a C, because remember in mRNA there are no T's, so you have to use a uracil instead. The T's are found in the DNA. So I'm going to show you some examples of this. But once again, here's that term codon. Codon is the group of 3 that you read in the RNA. Triplets are the groups of 3 that you read in DNA. So here's our DNA molecule. Let's say that this section here, there's a gene. So here's a gene. We're going to open it, and we only need to read one side of it. So you see this side says A, C, C, A, C, C, G, A, G, T. Well, then what we do is we're going to take that DNA, convert it to mRNA, and that's called transcription. So A goes with U, because remember, you're now in mRNA, and there is no thymine. C with G, C with G, A with U, A with U, A with U, C with G, C, G, G, C, A, U, G, C, T. Is that okay to be there? Yes, because that's in the DNA, and T goes with A. A is okay to have in the mRNA. So you're just using the base pair rule. Just remember, there should never be any T's down here in the RNA. You're going to replace it with a U or a uracil instead. Now what you do is you read these and you figure out which amino acid to be brought in. The amino acid, you read that group of 3, then you'd read that group of 3, then you'd read that group of 3, then that group of 3. So those groups of 3s are called codons. So this is a codon to bring that one in, this is a codon to bring that one in, and so on and so forth. So this is something you're going to have to do on a test. If I give you a DNA strand, I expect you to go all the way to build a protein, and I'm going to do a practice problem with you to show you how to do this. Now when you look at the code, which is taking that RNA and trying to figure out how to bring in which amino acid, As I said earlier, there's 64 different codes. 61 of them code for amino acids. 3 of them are known as stop codes, or they terminate the process of translation. So there will be 3 that you don't actually put an amino acid down for. They're just called stop. It knows right there at that point, stop building the protein then. Now, there is a special one that's a codon called AUG. AUG is the start codon, so it's the start signal to begin the process, and it also codes for a specific amino acid called methionine. So since AUG codes for methionine, and that means start, essentially every protein in your body, the very first amino acid is methionine, because that's always the start code. Now, if there's 61 different combinations, 64 because remember 3 of them were stop codes. However, there's 61 to code for amino acid, but there's only 20 amino acids. So realize there's redundancy in the genetic code. You can have more than one codon that can code for a single amino acid. However, there's never any ambiguity or any confusion. No codon codes for more than one amino acid. Each codes for one specific amino acid only. And I'm going to show you a chart and kind of re-explain that to you because it makes more sense when you see it. All right, here's something that I expect you to do on the exam. So obviously we'll walk through this. I'll show you how it's done. I want you to make your own up. You can make your DNA sequence whatever you want. Just put a whole bunch of T's and G's and C's, down in a row and figure it out. A, T, C, G, A, T, G, 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 T, A, whatever you want to do. And then you should be able to go from DNA to mRNA to RNA and then RNA to a protein. So we'll walk through it, do some practice problems. If it doesn't make sense, reach out to me and I'll go over it with you because you will have to be able to do this on the exam. So in your DNA, if you have T, and remember we're in the nucleus, and what we're going to do is convert to mRNA, and that conversion to mRNA, this is called transcription, right? So we're doing transcription, which is when we go from DNA to RNA. Remember down here, RNA to a protein, this is on the ribosome, and that one's called translation. So transcription and translation are what we're looking at here. DNA to RNA is transcription. RNA to a protein is translation. So if there's a T in the DNA, what do I put in the RNA? A. A goes with U. C goes with G. G, C. T with A. T with A with U. C with G. C with G. T with A with U. T with A with U. T with A, T with A. So that's what I would expect to see written out. You're just doing the base pair rule and matching up the DNA nucleotides with the RNA nucleotides. Now what we have to do is figure out how do we get these to a protein, and this takes a special way to do it. You have to find the codon chart and figure this out. So let me show you that. So this is what the codon chart looks like, and what it gives you is all the different possible combinations of your groups of 3 in mRNA, which once again is called the codon. So these are the codons you're looking at. So for instance, when you look at the codon UUU, that codes for the amino acid phenylalanine. When you look at the codon CUU, that codes for the amino acid leucine. I want to go back and point out something. Remember, there is redundancy in this chart. So if you have something like leucine, what can code for leucine? Well, CUU can, CUC, CUA, and CUG can all code for leucine. However, if you look at CUU, yes, there are other things that can code for leucine. However, CUU is not found anywhere else in this chart. It can only code for leucine. CUA could only code for leucine, so there's never any overlap in that way. One codon will only ever code for one specific amino acid, and so on and so forth. Now the other thing, here's a special one, there's that AUG, that codes for MET, which is methionine. It's also called the start codon, so that's why it says MET or start, because it is the start codon. And then here's those 3 stop codons. So UGA, UAA, and UAG are stop codons. So once you build your RNA, now what you have to do is say, okay, well, that codes for that, and that codes for that, and that codes for that. So how are you going to be able to do this? Well, if you ever go on and take something like biochemistry, you memorize this. Obviously, at this point, we're not there yet, so this chart will be on the test for you. You just have to be able to use it. So here's the problem we were working on before. You have your DNA strand. We already converted it into RNA through the process of transcription. And remember, that was just taking T to A to U, C to G, G to C, so on and so forth. So now what we have to do is translation. We have to take that RNA, convert it into a protein. And this, of course, is going to happen at the ribosomes. Now, this will be on the chart or on the exam for you, this chart. So what you have to be able to do is just use it. So once you build your RNA, now you look at those codes in your RNA to figure out what amino acid do I need to build my protein. So for instance, my first code, which is a group of 3 called a codon, is AUG. And you look around until you find AUG. That stands for methionine. Now, you don't have to know the full name of the protein. You don't have to tell me methionine. Just write down what it says, MET. Now we go to the next one, CAA. You look around your chart until you find CAA. So we found CAA there, and that's what it codes for, GLN. And then, so you don't have to know what that is once again, but that's glutamate. And then we have UGG, so we look around until we find UGG. There's UGG, and that codes for TRP, which is tryptophan. Then we have AUA, so we look around our chart until we find AUA, and that codes for isoleucine. And then that's an L. So isoleucine, just write what you see in the chart there, that's fine. And then we have a UAA. We look around the chart until we find UAA, and that's a stop codon. So you don't have to write stop. If you put it there, that's fine. I'm not going to mark it wrong, but that just means you're done. So here is our protein. Remember, a protein is a chain of amino acids. It's methionine, glutamate, tryptophan, and isoleucine. That's our chain of amino acids. And that's what I expect to see in the test. If I give you this, you build this, and then using this chart, you build this. That chart will be provided. You do not have to memorize it. The amazing thing about the genetic code is that it's shared between all living things that we know of anyway. So from bacteria all the way up to the most complex plants and animals. If the genetic code is found in one organism, that genetic code can be transcribed and translated by another organism. So you can take any gene, insert it into another organism, and get them to do whatever that it says. So we can program bacteria to make things like insulin for medical use. We have a lot of important biotechnology applications we can use this. That language that I just showed you is shared by all living things, and it's been operating since early life. Now, the amazing thing about that is, once again, you can take a gene, put it in any other organism, and make them do what that says, and that's the basis of what genetically modified organisms are. We can grow crops that are resistant to disease. At some point, we can basically make designer whatever we want to. You might have heard of the idea of making designer babies. Well, there's a whole bioethical, dilemma that goes into that, but in theory, we have the capacity or capability of doing that. So we can take plants, we can insert them with the firefly gene and make them glow. We can take animals, insert them with the firefly gene and make them glow, like this pig, how you see it has a glowing snout. Or these cats that can glow in the dark. We can genetically manipulate literally anything by taking one gene, inserting it in another. Because if you look at the idea of fireflies or bioluminescence and being able to glow, it's a chemical reaction. There's an enzyme called luciferase, and if you tell an organism how to make it, it has the amino acids, it just has to assemble it. It's like if you know how to make a cake, and somebody gives you a recipe for cupcakes, you should be able to make them. You just need the recipe. And that's all you have to do. Provide that recipe to a different organism, and they have the ingredients. They just have to build it now.